So welcome everyone. So today we'll have the first of Richa's talk. So welcome Richa. Professor Richa Rikhy is a professor at the Department of Biology at ICER Pune. She's an expert in early embryogenesis of Drosophila, where she's worked, I think, for decades now, right, Richa?
Not decade, but okay, a decade.
A decade now. So Richa did her PhD from TIFR, and she was then a postdoc at the National Institute of Health in Bethesda. And she's been at ICER Pune since 2010, where she now chairs the Department of Biology. And she also won the Senior Fellowship of the DBT Wellcome Trust recently. So Richa, today's talk is a research talk, I think. It's going to be the first part, onset of epithelial-like cell shape formation, and then tomorrow's talk is going to be the more pedagogical talk. So Richa, please.
So I'll decide what tomorrow's talk is based on the kind of interaction I also get from the audience. What I thought I could do in these two talks was, of course, show some very fundamental stuff of how, let's say, cells form and how this is interacting with gradients. So hopefully the interaction with gradients can come tomorrow. And today I will talk to you about the very first steps of cell shape formation, where if, for example, when we think about embryos in general, a lot of embryos will be either spherical or they will be elliptical, right? They are single cells, and then the first sort of morphologically distinct cell type which is formed is an epithelial cell. And I know that you have also had some discussion on cancers and things like that. So hopefully there is already a view of what an epithelial cell looks like. And I'll go through some, basic things about that. Now, when we think of organisms, they arise from this sort of a single cell. And we can imagine your favorite organism here, okay? Mostly, we've been studying animal cells through either literature or, experiments. But even one could think of plant cells, right? Where fertilization happens and there is a diploid zygote which is produced that gives rise to the embryo. And if then we start thinking of different organisms on the planet, they have a variety of different shapes, right? And of course, these are complex shapes which arise because of how tissues are arranged in these organisms. And we are really looking at fully developed organisms here for shape. But in all of this, the word that I'm interested in a lot, which comes again and again when these kind of transitions take place, is called morphogenesis. Morphogenesis, which essentially means the formation, the biological process which essentially gives rise to the form of that organism, right? Now, I'm interested in really early embryos, and we can look at any early embryo, zebrafish, mouse, and some of the simpler early embryos. And what one sees is that, by and large, like I said, they are spherical. And then, when their first divisions start, you start seeing some sort of interesting cell shape changes that take place. So, for example, this is a mouse embryo. And what one sees here is that if we can visualize one cell at a time, then initially they are very, very spherical. And then there are these protrusive structures which develop on one side. This is typically the side which is facing the environment. So imagine an embryo at a single cell stage. The plasma membrane of that entire cell is facing the environment, right? So imagine this one cell now dividing into two. So you have now two sets of environments which are occurring in this kind of cell type, right? So when it divides into two, the plasma membrane which faces outside is going to get cues from the outside environment, and the plasma membrane which faces the neighboring cell is actually joined with the neighboring cell, right? So these this is the first time there is a distinction of the environmental cue which the cell sees in a polarized manner, right? So, for example, this is the membrane which faces the environment, gets cues from the outside environment. This, on the other hand, is the membrane which is in touch with the neighboring cell and often has proteins which sort of make connections with the neighboring cell. So, so if we, for example, look at the mouse embryo, then 3 divisions take place. There is an 8th cell stage which is present. The cells are very rounded at that time. And there is a process which is called as compaction. This process called as compaction gives rise to a change in shape of this circular cell to a more distinct morphology, where if I start looking at the cross section of these cells, you start seeing polygons arrive, right? So a sphere or a circular cross section to that of a polygonal cross section. And you will appreciate that this polygon cross section arises because it has to come about because this cell and this cell have some proteins which are sort of stabilizing this membrane and really making a straight presence in this kind of membrane, right? So this transition is something which we really like to study. I also want to add, you just interrupt anytime. I'll start like this and I'll show you this transition stage in Drosophila embryos, right? So that is what I plan to do. Now, for example, if one looks at a schematic like this, so this is the 8th cell stage which we are talking about, and this is the compacted 8th cell stage. It's also what is called as polarized. Polarized meaning that it is now showing the presence of molecules differently in this membrane as compared to this membrane which faces the environment, right? So if we, for example, look at this membrane, it has some of these famous molecules. One of the famous ones, which I'll keep mentioning again and again, is ecadherin. And ecadherin is a transmembrane protein. This transmembrane protein now has an extracellular domain which binds to the extracellular domain of neighboring cells. So it's sort of like they're holding each other, right? And this requires the presence of, for example, ions like calcium, which is what stabilizes this kind of association which is present. There are others, we won't go into those right now. The apical region often has a lot of these villi-like structures, all right? So these are essentially protrusions from the membrane. These protrusions, essentially, they are serving what is happening in the milieu or the environment around the cell, and they are also thought to be very important for increasing the surface area so that a lot of contact can happen between the apical membrane and the outside portion of the cells. So, of course, these are protrusions and they have to be stabilized, and oftentimes they are going to be stabilized by actin filaments right here and actin filaments at the base as well, which consolidate their presence. So, hopefully this can already, put some impression on what is the first specialized cell type that develops in embryos, and it's an epithelial-like cell. I'm still not calling it an epithelial cell because this still does not have all the components which are very, very similar to mature epithelial cells, but it has all the features of an epithelial cell, right? So what are the features of an epithelial cell? When we started looking at this as well, one of the ways of doing this is to go back to mature epithelial cells. And it really turns out that if one looks at mature epithelial cells, so where are these cells found? These cells are essentially present as a lining to many of our organ systems, okay? And when one looks at these cells, they are compactly arranged next to each other. They bind each other, and these binding regions occur in different forms all along these membranes. So here is an apical membrane, which is now shown as a straight membrane, but often has villi-like structures. there's a lateral membrane, and here this lateral membrane has adherence junctions, which has the same eq-adherent molecule, which was shown there. And it has other polarity complexes, which we won't really go into as much right now. And then there is the basal membrane, which contacts the substratum. Okay. So we have all of these different membranes, which not only help to hold tight the neighboring cell, but they also bind to the basement membrane. And when we look at the cross-section of these cells, we see this sort of a membrane organization where if we start counting the number of edges which are present, then you will very often see a distribution between 5, 6, 7, 8 sided cells. All right. And if we look at the distribution of all of these edge numbers across many different organisms across the animal kingdom, then there is a predominance of the hexagon which appears. All right. So what is the meaning of this hexagon dominance? And I mean, for example, in beehives, and I'll show you a picture of that, a very stable beehive will usually have a hexagon dominated organization. Where does it have pentagons? It has pentagons at the edges. All right. So a distribution between hexagons and pentagons is needed to maybe have curved shapes. So for example, if any of you know about a football, what does a football have? Anyone? Yeah? Hexagons and pentagons, right? So it's a combination of hexagons and pentagons. If it had only hexagons, you would mostly have a very sort of planar appearance. So there's a reason why this has hexagons. Anybody thinking of what the reason could be? Why is hexagon something which is seen so much and again in all of these different animal types? Sorry?
Tubular structures, maybe?
So you're talking about the entire shape of the cell, right? So essentially, right now, one way of explaining it is that the hexagon architecture is a very energy minimized architecture when it comes to surface area to volume ratio analysis. All right, so it's the best structure which will fit in this kind of regime with the most optimized surface area. All right, so one could think of filling this with squares, you could fill it with triangles, but one of the best ways, the most optimal way to fill such a system is a hexagon. Now, do we want a hexagon structure all the time? No. Okay, no, that is also not okay. So this is an example of that. Again, the same figure just enlarged here. You will look at these two kind of sheets. This sheet has hexagons and this sheet has, all kinds of different shapes. And you can see the cells are jiggling. They are not going to be, frozen. They are not in a statue state, but they are sort of moving around. And here what you see is that there is a change in edges which is coming about, especially when there is an interesting migrating cell going across. And there are ways of defining this. This is a very stable structure. It is jammed. And this is a structure which is more plastic, where there are changes taking place in the number of edges, and it is, called unjammed. And what we got interested in trying to ask was, can we figure out whether these kind of shape changes are coming about in, all kinds of embryos? And the embryo that we started studying was the Drosophila embryo. Now, Drosophila, the same insect species which you will find on bananas. And for various different aspects of biology, research in flies has been seminal in making us understand how, gradients form, how cell shapes change, how behavior happens for many different wild-type behavior, like circadian rhythm is an example. We are looking at essentially early embryo development. Now, it has a peculiar embryo development, all right? insects, all of them, they do have a peculiar embryo development. So unlike the mouse embryo, which I was showing you earlier, where each cell, when it divides, forms completely two independent cells. They are bound to each other, but they are independent cells. Here, what happens is that the first few divisions occur internally inside the center of the embryo, and these are essentially nuclear divisions. And after a few cycles, which is 8 cycles, most of the nuclei migrate to the periphery. When these nuclei migrate to the periphery, along with the nuclei, there are other kinds of substructures which are going to migrate along with the nuclei to the periphery. One of them, for example, is the centrosome, which also will come to the periphery. And the centrosome and the nuclei together make an organization of the plasma membrane such that there is like a half cell. It's like a protocell which forms there. But this protocell is not a complete cell, all right? So there is no membrane at the bottom. And then there are these cortical division cycles which take place, and it's only after many division cycles which fill this entire surface. So in this case, it is essentially 13 division cycles. In the interphase of 14, there are 6,000 nuclei which are actually present at the periphery, and around these nuclei, the plasma membrane pushes down and forms complete cells. All right. So now these are full cells and these are epithelial cells. All right. And all of these cells are actually showing these binding patterns of membranes adjacent to each other. Right. So like, for example, cadherin is present right here. Now it turns out that, when many scientists started studying this from the very beginning, what they saw was that not only in this stage where complete cells form, but even in this stage when there's like a half cell, if we look at the cross-section of the cell, that also actually appears polygonal in nature. Okay? And so what we thought we could do through some of our work was understand what are the geometric principles which are operating to give rise to maybe a polygon-like architecture in these cycles. All right, so I'll show you some movies to maybe illustrate this point. Now, so this is a more textbook picture of this, where you see nuclei in the middle. Not all the nuclei go to the periphery, some remain in the center. These ones which are present at the periphery, if we zoom in, this is how it looks. There's a nucleus. This nucleus actually has all the organelles present around it. So there's endoplasmic reticulum, there is microtubules, there's a lot of cortical actin, and so on. So it's a very crowded system. It has interesting mitochondria which are associated with each cell. And it turns out that even though this membrane is not extending and making a full cell, it's not like, mitochondria which are present here, or the Golgi complex which is present here, is really wanting to go to the neighboring cell. It doesn't go there. All right. So the reason it doesn't go there is because all of these organelles are sort of linked to a very organized cytoskeleton system. And this is the microtubule cytoskeleton system, the actin cytoskeleton system. So the organelles are very important for their function in one domain. And we are calling it a domain because it's, by definition, a cell has boundaries, right? So in this case, the boundary is not the plasma membrane, but this entire set conglomerate of things, which is the cytoskeleton, the nucleus, the microtubules, and so on. So this entire set of things essentially forms what is called as the nucleocytoplasmic domain, which is one cell-like feature of the system. Okay, so when we think of divisions over here, so we have these incomplete cells. We look at the cross-section of these cells. We can see that at some point they are actually circular, and that matches to some stage of the division cycle. And at some point, they transition from circular to polygon. And so imagine there's this beautiful entire array of cells right here, which is all together in a synchronous manner, transitioning from a circular cross-section to a polygonal cross-section. Okay, and the size that we are thinking of right now. So in case of embryos, one other thing which is conserved from the very beginning is that they have a fixed volume in which all of these divisions have to happen. This expansion of volume does not occur for quite a while. All right. So in this fixed volume, you can imagine as the nuclei keep dividing, each cell-like entity is of course becoming smaller and smaller and smaller. Yeah.
You see in this interface and this prophase, there's this empty space. It's like just empty space, or like there when the cells, circular cells are joined, right?
Yeah.
So there is this empty space in between.
Yeah, so this appears like an empty space. It's just a little space which is actually present in between adjacent caps. So when we, for example, so at this point, if you take this cross section, you see this empty space essentially. It's this empty space, and if you go inside and inside, this empty space is essentially going to show you membrane, which is more polygonal in nature. And this is not really empty. So for example, when we look at the entire embryo, if this is the plasma membrane, it is this perivitellin space. So this is the white line membrane, and what I've not shown you is a third cover of the embryo, which is the chorion. Okay, so this is a space between the white line membrane and the plasma membrane, and it is filled with perivitellin fluid. That perivitellin fluid has a lot of important cues. They have, for example, factors which actually activate some of the morphogen gradients. All right, so it's not empty space, but yes, it is space where we have this kind of perivitellin fluid. Yeah.
And when it takes on to the form of this polygonal shapes, this fluid, it goes away.
Yes, absolutely. So it's kind of getting excluded, right? Yeah. So the imagination is absolutely right.
This also happens.
And I'll show you a movie of this as well. Yeah.
Is it because of the pressure gradient, like pressure difference?
Is it because of the pressure difference? So at this point, you could ask this in another way. Is it because now the membranes are actually contacting each other so tightly that this little edge, edge of contact has now spread so that it becomes a polygon? So certainly there is an exclusion of the volume. And hold this thought on the pressure, and we'll come to a discussion on what are the changes in mechanical properties of this kind of system, right? So since you asked the question, it's also interesting to think of how is a cell stabilized in this kind of circular conformation, and then how is this circular conformation transitioning to this polygonal conformation. And since I'm a biologist, my interest is to try and understand what are the molecules which are important for making this kind of transition, and what is the geometric distribution of this polygon which is actually present in these systems.
Certain role in making polygon structure, because
absolutely.
we have to separate.
Yeah.
Then maybe.
So this is a very nice question. What you're asking is whether it is this. So what happens is, along with this, there is also a transition of the lateral membrane, right? So of course these cells do not have a basal membrane, but that's okay. What they do have is lateral membrane, and this lateral membrane is increasing in length across this division cycle. All right. And this lateral membrane is very similar to a lateral membrane of an epithelial cell. It has, for example, the adherence junction molecule E-cadherin, which is actually present right here. And E-cadherin, in fact, is the most stabilized in this particular state. So the metaphase plate really is right here. This is the plate where the DNA is present. What is present towards the periphery is the centrosomes. The centrosomes are the ones which emanate the microtubules, and they stabilize the spindle. the centrosomes play a key role in helping this maturation, if I can call it, of the lateral membrane also, right? So at the same time that the cross-section shows you the transition of shape, there is an increase in the lateral membrane which is ongoing as this shape transition takes place. And hopefully what you've noticed now is that we can use this system to understand what is the need or what is the length at which this kind of transition takes place. Of course, what is very clear is that when this lateral membrane is small, this particular organization is circular, and when it is longer, the organization is polygonal, right? So this transition might depend upon the length of the lateral membrane. And what is length? Biologically, length is the sum total of proteins which are getting associated in this kind of membrane, right? So let's say there's X amount of protein which is stabilizing the lateral membrane here, and maybe there is 5X amount of that protein right here, which is helping for binding these two adjacent membranes and therefore stabilizing a polygon-like architecture. Yeah.
Well, say now I make these cells and actually spread them in a free space, say in a petri dish or something, will it turn to circular cells?
So I will show you a way of transitioning them into circular cells. So at least, we should hopefully reach that part of the talk. So also you must think about when this is circular, think of also what are the forces inside this cell which are keeping it circular, right? And these forces have to go away if this structure becomes a polygon. And typically when we are talking about these forces, they are of course cytoskeletal forces, right? We are essentially talking about the cortex of the membrane, and this cortex of the membrane is obviously rich in the actin cytoskeleton. The actin cytoskeleton is what is having, let's say, a certain rate of contractile forces to keep this circular architecture. And if only that circular architecture or the contractile force that maintains the circular architecture is now silenced or is abrogated, that is when, a more polygon-like structure will form, right? So it's also interesting to think of what maintains this shape and then what transitions it into this shape. And really, a balance between the contractile and the adhesive forces is something which is responsible for changing shape in almost all organisms. All right, and this is not something which is being found for the first time here. It is true for every epithelial cell type, right? So this is what we thought we could study in relation to the kind of cell shape changes which are taking place right here. And I don't know if you saw a lot of movies of, tissues remodeling, but GFP really changed our ability to try and understand not only how proteins distribute inside cells, but also how cell shape change in development, right? So GFP from the jellyfish, we can tag this to any protein of our interest, make a fusion construct, put it into the embryo, and then look at how these cell shapes remodel. And here are 3 different examples, okay, of 3 stages of development. So don't get, focus on all the stages at the same time. Focus on this, for example. These are those early division cycles. Here, what is labeled is the histone. Histone essentially binds DNA and marks the DNA. And what you see here are almost synchronous division cycles, and they are essentially leading to doubling of the nuclei, right? So you're not seeing the membrane here. I'm going to show you the membrane in these stages in the following movie in the next slides. After this stage, after this syncytial stage, there are 6,000 nuclei which are present right here. Now, this movie does not show you nuclei. What this shows you is the plasma membrane around every nucleus, and you're looking at the side view of all the cells present in one row, where in a fantastic process, this membrane drops down around each cell and forms what is an epithelial cell, right? So now these are 6,000 epithelial cells which are actually forming in a matter of about 40 minutes. And they are epithelial in nature, they are polarized. So this is a cell formation process which is taking place. And so together, because this is cell formation, it is polarization, this process is essentially called as cellularization in Drosophila embryos. And what you saw here, right, this is the time point at which the first germ layer formation takes place. So up until now, at the end of this, you essentially have a full embryo which has one sheet of epithelial cells. And at this point now, there is a cell shape change which is taking place, which allows for a bunch of cells in these regions to get apically constricted. They constrict from one part of the membrane and they go inwards. And this time point at which the cells go inward, now you can imagine that it's the first time that the embryo has two cell layers. One which is on the outside, which will finally form the ectoderm, which is, for example, going to form things like our skin. And the other cell layer, which is gone inside, which is going to form the mesoderm cell layer, which is, for example, the muscle sheath. And this particular process is also akin to the epithelial to mesenchymal transition, which takes place, and you don't want it to take place, for example, in cancers, right? So you don't want that transition from epithelial cells to mesenchymal cells to take place in a fully formed adult organism. This is something which occurs routinely in the formation of our tissues, right? And it is needed in the formation of our tissues. And it is, of course, needed the first time we give rise to or have two different cell layers which are present. So, in principle, one could sort of break down this question of early cell shape changes into three kinds of questions which we studied while actually just looking at this in detail in terms of what kind of morphologies are changing, right? So, one could look at the apical membrane. This is, so apical meaning membrane on the top, and when one looks at the apical membrane, it is not just a circle. It has a lot of protrusive structures. It has a lot of villi-like structures. So this is what it is present when it is circular, a lot of villi-like structures. And when it becomes a cross-section polygon, it's exactly a polygon. There are no villi-like structures present anymore. And when we look at the distribution of these polygons, then what you see is that you indeed get a varied amount of distribution of 5, 6, and 7 cells. Alright, so this is essentially three things which we have been studying in the lab, and we sort of have some information about each of these steps. And what we are interested in is in trying to understand what sorts of molecules regulate these kind of transitions, or the formation of a more stable, let's say hexagon-dominated polygon architecture in the early embryo. So, for example, if I'll show you some things about this. Okay, so maybe I'll cut short. I'll show you just a few things so that we can have a discussion rather than, me showing you data. So, for example, when we look at this early stage, this has not been seen by us for the first time, okay? This is known for a long time. And when one, for example, uses electron microscopy to look at the surface of these cells, what one sees is these circular caps and, the gap which someone at the back was alluding to, this is essentially the gap. So it is because there is a bulge, right? And this bulge, it has these protrusive villi-like structures, and in between these caps, there is a gap which is most likely filled with the perivitellins fluid. When we look at this using microscopy, labeling some of the villi-like structures with a marker which labels actin, and we can look at, different division cycles. So when the 10th division cycle happens versus when the 11th division cycle happens, 12th and 13th, and so on and so forth. What one sees here is that, so basically what you're seeing here is this cap-like structure, and hopefully you can already appreciate that this cap-like structure is big. Then this one maybe is roughly half of this, and this one cap-like structure is roughly half of this. And in cycle 13, they are smaller caps, right? So in the same volume, we are seeing that the caps are becoming smaller and smaller. There's increased number of caps which are actually present. These are rich in these villi-like projections, which we can see better with a slightly better microscopy method. And when we, for example, look at different stages of the division cycle, what we see here is that in the division cycle in metaphase, there is a more polygon-like distribution in these cycles, and you do not have these villi-like structures which are present at all, right? So obviously, all of them are going away. And most likely, all of them are going away and forming the lateral domain, right? And so this is a very nice, interesting remodeling step which one could study to try and see how this transition is actually taking place in these stages. If we look at them carefully, for example, in the edges of these caps, right? So this is one cap, which I was drawing very naively, just like a circle. There are these protrusive structures which are present. They are as if they are meeting the protrusive structures of the neighboring caps. And finally, they will match up with each other and form a polygonal structure. All right? And so one could, for example, quantify how many protrusive villi are present. One could quantify how many are present, or what is the length of the villi and what is their number in different stages of the cycle. And what one can see is that there is a decrease in the length of these protrusive structures as we change the cycle, and there is also a decrease in the number of villi. And what's happening to these villi, or the membrane which is present in these villi, it is basically forming the lateral domain. Okay, so it is forming this structure, right? So there's a transition which is occurring from this circular structure to a more polygonal structure. Now, what does this polygonal structure depend upon? Okay, and so this is a movie which allows you to see this kind of transition. So what are you seeing here? So imagine this embryo. It is lying on a cover slip, and we are seeing it in the microscope. It expresses a fluorescently tagged protein. That fluorescently tagged protein is going and associating with the membrane of these embryos. And we are looking at only a small portion of the embryo. Okay, so if I want to give you dimensions, we are essentially looking at maybe something like 1/20th of the embryo. Okay, and so we need to zoom in to a particular region so that we can actually see the cells dividing. And when you look at this particular section, this is essentially looking at the embryo down, right? So imagine that the embryo is just sitting in front of you. You are looking at it on the surface. And what this is allowing you to do is to look at the cross section, right? So this cross bar is where you are looking at the membranes inside the embryo. Okay, so now when this movie starts from the beginning, what you will see is that there's a division cycle which increases the number of these protocells which are present, and another division cycle which increases the number of cells, and another division cycle which increases the number of cells. So this is the 13th division cycle which takes place. And now notice what's happening in this lateral membrane, right? So there is a cycle of membrane ingression, regression, ingression, regression, all right? So there is this ingression and a regression which is taking place at the same time. And what we wanted to ask was whether the change in shape which is occurring in these stages has anything to do with the length of the lateral membrane in question, right? So we can sort of systematically look at this. If we look at static images along with the nucleus, then we can see that at interphase we have the nuclei which are present. At metaphase, there is this metaphase plate where the chromosomes are organized. The shape is more circular here, it is more polygonal at this point, right? If we look at the extent by which the membrane is extending, right? So this is the lateral membrane. Now in that same time period, what we see is that more and more and more lateral membrane is forming. And obviously this is most likely correlated with more and more accumulation of important proteins which are binding these lateral membranes to each other. If we look at how the cap is remodeling, so here is one cap, here is the cap in a later stage of the cycle. What we see is that the cap area increases, it stabilizes. And then what we see with the furrow length is that it increases to a maximum and then it decreases again. All right. So this is the phase that we are interested in. So here is where the cap area increases and stabilizes and also gets rid of all of the protrusive villi-like structures. All right. So this is the cell shape change which I was talking about. Now if I look at just one cycle again and again and again, what I've done is I've drawn these yellow regions for outlining the cells. Okay. What we see here is that there is a very interesting stereotypic repetitive transition which is taking place where the boundaries are fuzzy, okay, that is when the villi are present, and where the boundaries are more taut, they are tight, and that is where the polygon structure is present. So just to point out, fuzzy boundaries and taut boundaries, right? So this is exactly transitioning in cell shape. The interesting thing is that this cell shape transition is occurring in a matter of about 10-15 minutes. That's it. So it's a very nice process to be able to visualize as well as to try and understand whether the rate of this transition takes place properly if I start removing some important proteins which are responsible for this transition. So just looking at the wild type, just looking at, wild embryos without making any perturbations, what we saw in this movie was that you start with somewhat of a rounded cell and you become a polygonal cell. If we correlate all of these stages with the lateral membrane, what you can see is that there is an increase in the lateral membrane boundary which we see. If I can, measure this kind of change of shape by a mathematical factor, this is so-called circularity, and essentially if it is completely a circle, you will get this at 1. What you see is that this is dropping down to when the polygonal architecture forms, and no matter which cycle I look at, right? So I can take the 11th cycle, 12th cycle, 13th cycle, doesn't matter. There is always this drop down of circularity to a polygonal number, and this occurs at the same rate. And when I correlate this to the length of the membrane, there is some sort of very interesting threshold which is coming up. All right. So which means that when, for example, I am at this kind of length, about, let's say, 6, that is when the shape change has already occurred. Right. So what is the meaning of this? Right. So I don't know if I'm losing you, but what I'm trying to understand is what is the extent of this lateral membrane which needs to be present for this kind of shape change to take place. And why is that important? That is important because it has, hopefully, along with the length, some extent of protein signature. So maybe, let's say, the amount of DE cadherin which is present, which can zipper those membranes enough and stabilize them to form this circular to polygon architecture, right? So there is a lot of interesting changes which are occurring from this circle to this polygon, right? So when we think about this, we also can immediately think about what is the change in mechanical properties which is occurring right here, right? So when we are thinking of mechanical properties, we are thinking of a balance, very, very broadly speaking, between two molecules. One of them is cadherin, which binds these two membranes. The other is the actomyosin contractility. All right. So if the structure was very, very contractile, right? So imagine that inside the circular membrane, if the structure was very, very contractile, then you would actually not see this transition occur at all. So contractility has to reduce, only then this kind of transition will take place. Okay? This is essentially this lateral membrane length, this length. It's called a furrow because it's a furrow which is going into the embryo.
So on the green line, it seems to be increasing in circularity. So
Yes,
is it
at this
because
point,
of
right?
another cell cycle?
No, it's a nice observation. The reason why this is increasing is because the number of hexagons here is more and more and more. And I'll show you that. So when for this factor essentially is 1 for a circle, and there are a lot of pentagons which are present at this point, and when the number of hexagons also start increasing, then there's a lot of fluctuation here, which is much more closer to a circle than, for example, in this particular stage where there are more pentagons which are present. This will maybe become a little clear when we sort of start looking at the distribution of the polygons in this kind of architecture. Okay, so what is the meaning of the hexagon, right? So typically a lot of hexagons are present in beehives, and in tissues mostly there's a balance between hexagons, pentagons, 8-sided, 3, 4, 5, 6, 7, 8. Okay, and depending upon how plastic the tissue is, there is a distribution of all of these which takes place. What creates this balance is also a balance, very broadly speaking. It's not these two factors. I'm really oversimplifying the whole situation. But two factors. One is this E-cadherin, which has this feature of binding to neighboring molecules of E-cadherin. And the other is this contractile cytoskeleton inside the cell. So now you can imagine that if this was super contractile, right? So inside this, if this is hypercontractile, then the entire system will really become like a circle. If it is less contractile, then it will allow for this adhesion to take over, right? So one of the things which one can do is then ask whether when I increase the contractility, how will the shape change occur, right? So one could also do a very simple thing, is that while this cell shape change occurs, one could look for what is the distribution of E-cadherin and myosin. All right, so this is myosin. It is a part of this contractile cytoskeleton, right? So this is what is giving rise to bundling or overlap of a lot of actin filaments. So actin filaments are filaments of the cytoskeleton. Myosin is a motor protein which sort of makes them glide against each other and really create the contractile force. Now, if there is more contractility, then this adhesion will not be stabilized very much, and you will see a more circular system. So first thing one could ask is, we already know that the cell shape change occurs, right? So we know that, for example, to begin with, they are more circular, and then they become more polygonal. Now, what you can hopefully appreciate in this color scale is that, as it goes away from the very blue color, there's more and more E-cadherin which is present in these stages as they become more and more polygonal. Okay, so more and more adhesion is actually happening. And when we look at, for example, this myosin, this is one of the molecules which are part of the myosin complex. Then what you see is that there is a lot of myosin which is present here when it is circular, and not surprisingly, it is all gone when it is a polygonal structure, right? So this is an interesting correlation. It is only a correlation that when this shape change is taking place from circular to polygonal, there is an opposing distribution of myosin and ecoherin, all right? So the contractile nature is decreasing and the adhesive nature is increasing, okay? And I mean, I could, sort of transition from here into many different places. What I thought I could do is in the next, let's say, 5 or 10 minutes. Yeah, it's about 3 p.m. now. So what I thought I could do is try and think a little bit more about how we can measure the contractility or the tension in this kind of system, right? So imagine that it is more and more contractile. Then what one will end up doing is one will of course change the shape of the cell, but one will create a membrane which is under a lot of tension, right? So as much as I can use these words to describe what the membrane might be feeling, are there methods which allow you to dissect these, right? So there are 2 methods which, for example, I would like to share with you. One of them is that, let's say, if you had a lot of myosin which is present here, right? So this myosin is going to be very tightly engaged with the actin cytoskeleton. It will not be roaming around. Okay, so can we figure out how much this myosin is roaming around? All right, so that method is called fluorescence recovery after photobleaching. Another method is, and I will show you what that means. Another method is, can I figure out how much tension this membrane is in under, right? So if it's really tight, if, for example, it's like a rubber band which is really stretched, then it is under a lot of tension. If I cut the rubber band, it will snap, okay? And it will snap at a speed dependent upon the tension in the rubber band, right? So that actually tells me how much tension is there. All right, so this is a brief introduction, and let's look at what this really means, right? So this is a method which is called fluorescence recovery after photobleaching. This is the method which allows you to understand how much are molecules roaming around in the cell. Okay, if a molecule or if you are busy talking to your neighbor, then you are engaged in talking to your neighbor and you will not be roaming around in this room. All right? Can I understand how much time a molecule is busy essentially in a particular region of the cell, right? So the way one could do that is, let's say I had a cell and I can label my favorite molecule, let's say ecoherin in this case, in the cell, or myosin, right, the contractile one in this cell. Then it will give me a signal akin to its distribution inside the cell, right? So that's what GFP did for the entire field of cell biology, where now we can visualize how the proteins are distributed inside the cell. I take a region of that cell where, let's say, the GFP protein is now very predominantly present, and I will do what is called as photobleach. Okay, so now this is a fluorescent protein. It will give rise to the phenomena of fluorescence at a certain laser power in the microscope. I can use a really strong laser power, and in that case, essentially what will happen is that in this region, the molecules will switch off their fluorescence. Okay, it's basically similar to switching off the lights. So that molecule is present, but it is not showing anymore. And if this molecule is also roaming around in the cell, you can imagine that in this region, I switched off the fluorescence. These off molecules are now roaming around everywhere, and this region is getting filled by the molecules which are coming from this place where they were not affected at all, right? So this is a method which is called fluorescence recovery after photobleaching. Photobleaching is a method of switching off the fluorescence, but the function is not abrogated. So the molecules will keep moving around anyways, and then they will fill this space. Now, what can we do with this kind of information, right? So this is a live movie of an intact cell. It is giving me information about the dynamics of the different proteins inside the cell, right? So, for example, if you imagine all the information that I gave you, it will give me information about the dynamics of the myosin. It can give me information about the dynamics of the cadherin. And it will give me information when the cell shape change is occurring, right? So we want to be able to get this information in the process of our interest. What can we do with this information, right? So you are able to make a region of interest, bleach it, that region of interest gets exchanged by these traveling molecules from this place to this place. What is happening is that in this region which you are monitoring, there is a decrease in fluorescence because of your action of photobleaching, a very high intensity laser which is put there and the fluorescence just switches off. And there is a recovery which takes place because of molecules in this region which go and flood this rate, right? So now this recovery or the rate of recovery is very, very informative. The rate of recovery tells me how fast are these molecules diffusing in this particular regime, right? And is this interesting? For example, if I knew how fast were molecules diffusing in this particular system when, let's say, the cell is in the circular conformation or when it is in a polygonal conformation, is this something which will tell me how the molecule is organized when it is circular? How is the molecule organized when it is polygonal? All right. The diffusion of molecule is going to be related to its position. The diffusion of molecule is going to be related to what are the molecules it is interacting with. So, where will I do the photobleaching? I will do the photobleaching exactly on the membrane. I will not do it in the cytoplasm, right?
So, like, if we do the photo bleaching on the membrane and the molecules are in the periphery of the membrane, so the diffusion will be fast for that thing. And if it comes from the center, it will be slower.
So it depends upon where it is, right? So now imagine, so this is a very crude diagram, right? So this diagram essentially gives you a region. This is a cytoplasmic region. Now, when you think of the cytoplasm, it is like a fluid cytoplasm. The fluid cytoplasm has molecules which are really roaming around very fast and they are not engaging very much with any one molecule, all right? But when the molecules are present in the membrane, they are interacting with each other, maybe with the neighboring molecules, or they are interacting with the cortical cytoskeleton. So they are very choosy when they are in the membrane. In the membrane, they are in a conformation where they are poised to interact only with a certain set of molecules, right? So it's a nice question. What it allows you to do is allows you to design a suitable experiment where you do this photobleaching in the cytoplasm or you do this photobleaching in the membrane. So what is your thought about this? Suppose I had molecules which were present in the cytoplasm and I had molecules which also had the ability to go into the membrane. What is going to be diffusing faster? The cytoplasm, right? And the ones in the membrane are in a completely different medium. The membrane is crowded, it has lipid, it has many other proteins, and it is going into that particular phase only. So now, for example, we did this for the myosin cytoskeleton, right? Using that one molecule which labels myosin. And we did this when it is circular, and we did this when it is essentially starting to become a polygon. Right? So, if you look at the recovery, so the recovery also tells you the extent of diffusion, right? So, if the recovery happens very fast, then you know that it is very fast exchange with this particular region which is photobleached. If the recovery happens very slow, then you know that the exchange is also very slow. If the recovery does not happen to completion, if there is a so-called immobile fraction, which is never going to be replaced, then you know that the molecules in that region are fixed. They are going nowhere. They are essentially just placed in that location. And only the mobile molecules are the ones which are going everywhere. All right, so this is a very, very informative analysis, which when done with different proteins and different regions in which the proteins are present, will tell us the extent of mobility of the protein itself. Okay, and so when FAN, for example, does this for a circular organization of myosin versus a polygonal organization of myosin, it's interesting to see differences, right? So this one is when the organization is circular, okay, and this one is when the organization is more polygonal. And can you see a difference? There's a lot of noise there. It's expected because also the embryo is a very moving structure. It's not a static one. But there is a clear difference which is coming out. In this particular point, which is the circular portion, you can see that the recovery is higher. Okay, if it would recover completely, it would go to 1, but it doesn't. If you look at this particular portion of the membrane, when it has just started becoming a polygon, it's not completely stabilized into a hexagon-dominated structure yet. It has just started becoming a polygon. There is a more engaged association of the myosin molecules with the cortex of the membrane. And so we can say that this change just correlates with the cell shape change. Okay, are we saying this is responsible for it? We don't know that yet. When will we know that this occurrence of myosin is responsible for the cell shape change? When will we know that this myosin activity is responsible for the cell shape change? We can directly measure the tension. We can remove myosin. We can increase myosin. All right? So all of these. And essentially what people want to do is all of the above. Yeah.
So in the previous image, we can see that there is in the membrane, the recovery happens quickly if we see with respect to time, but then eventually it sets up to a lower basically intensity, so that we can see that there is more of a mobile fraction in the membrane. But what does this implies? That the recovery is faster in the case of the membrane compared to the...
So at this point, we can only say that the way that the myosin is organized is different. All right. And what we can say, so for sure, we can say that the myosin is very intense here. You can already see that. There's less myosin here. When we look at the organization, what we can say is that there is more mobility for myosin in these stages, and there is less mobility over here. All right. So at some point, it is getting engaged much more, and therefore the recovery is less. Right. So again, going back to what this assay allows you to do is to look at the amount of protein which is present in the immobile versus the mobile fraction. If you have a lot of mobile fraction, you will have a lot of recovery which takes place. And when you have a lot of recovery which takes place, that tells you that the molecules are exchanging very, very fast. It's like you are roaming around this entire room all the time. But when you are sitting in one place, and if there is a way of, if you had a fluorescent molecule and I could switch off that fluorescent molecule, basically nothing will replace it. Okay, because everything is sitting in one place. And that is what essentially it says. It gives us some sort of an intermediate picture because nothing recovers back to 1, right? So in both the stages, there is both mobile and immobile fraction. What we can ask or find out from here is the extent of change. What we see here is that when it is in this state, there is this amount of mobile fraction, which is higher than when it is just beginning to become a polygon. So that tells us that the architecture of the cytoskeleton, of the actomyosin cytoskeleton, which is present beneath the membrane, is changing for us to give rise to this particular change. Okay? Yeah.
So you had this previous curve where you were showing that myosin itself is going down, right?
Yeah.
When you have this shape change. So is it that the immobile fraction, it's not that it's getting more engaged, but that
Yes.
number can remain the same, whereas proportionately it seems like it's falling because...
So over here, there's more myosin and it's exchanging a lot.
correct.
And over here, there's less myosin, but it seems to be more stable.
Right. So it's the relative fractions that are changing rather than the absolute
Yes.
number of...
So what is interesting is this point. This point has some intermediate amount of cadherin. It has some intermediate amount of myosin. And this is the point at which the shape change is occurring from circular to polygon.
And so just as a follow-up, so these mobile myosins, can they penetrate into the lateral membrane, or is it...
They can go anywhere
Okay.
once they are mobile. What we've observed for myosin is that they do not come to the lateral membrane ever.
I see.
So that lateral membrane, which is the most stable lateral membrane in metaphase, has only cadherin. It doesn't have myosin.
Doesn't.
I just want to clarify the observation. So when we are looking at the transition from interface to metaphase, so the observations are that the number of microvilli are reducing. Right?
Yeah. So it's, the same microvilli, they're getting stretched down.
Okay.
They're essentially getting remodeled. There are many different processes occurring, but one of the things which is occurring is that the villi are just getting stretched down. They're providing membrane for that lateral region.
Okay. So the microvilli, the number is reducing.
Yes.
Circularity is also reducing as you transition from interface to metaphase. So between nuclear divisions, so does it go back? So it's like circularity to polygon
Yes.
shape, then circularity to polygon shape.
Exactly. So it cycles.
Okay, right.
And so when we're looking at cycle 11, 12, 13, these three cortical cycles, in those cycles, there is a lot of cycling of shape which is taking place. So it's from circular to polygon, to circular to polygon, to circular to polygon. There is short membrane, long membrane, short membrane, long membrane, short membrane. Villi, no villi, villi, no villi, villi, no villi. So these very striking changes occur one after the other in these cycles.
Okay, so what is there a purpose for the villi to exist on?
Yeah. So many people ask this question: what is the reason to have those villi?
Yeah.
So one is that they are the reservoir for that membrane which extends downwards, and what else are the villi doing? So in other systems, and we haven't studied this in the system, and maybe it is also true for the system. In mouse embryos, this is also known. Where these villi-like structures are present, often sometimes at the tips of the villi, E. cadherin is present. So it's like, villi are coming close to each other, and this is where the E. cadherin is brought and it stabilizes to form a lateral membrane. All right, so there's some purpose of these villi to come together and form this kind of structure. So it's very possible that the villi play a very important role for not only acting as membrane reservoirs, but also for providing a place to park the cadherin so that it can meet the neighboring villi and form these consolidated lateral membrane. Hypothesis right now, okay? Based on literature from other systems, it's possible it is true, it needs to be tested.
It's that one.
Yeah, it's very coordinated. So it's like the two caps. So he's asking whether the villi coordinate their motion. The two caps the villi come towards each other in a very coordinated manner, because all the caps are small and they expand, and the villi are coming together in a coordinated manner, for sure. The coordination in part happens because of the way the division is itself taking place. So from interphase to prophase to metaphase, there are so many interesting changes which are occurring. One of the changes is also that the nucleus is becoming a little bigger, and the entire volume which is occupied by that system is somewhat changing in organization such that the cap spreads. So all of them are doing it at almost the same time. So that spreading is sort of synchronous in local regions. Okay, so what I can do is I can just show you this and stop after this. So one of the other ways of measuring tension is by actually measuring tension. Okay, and like I said, that if we think of tension as it is present in an elastic rubber band, then what we can see here is that if we, for example, didn't stretch it at all, then one would really have that rubber band in a low tension. If I cut it, you won't feel anything on the nails. All right. If I stretch it and cut it, you will feel a snap. Okay, and this already can tell you that when you had stretched the rubber band, it was under a greater amount of tension. So can we do an experiment akin to this inside the cell? Right, so imagine the actin-actomyosin cytoskeleton. It is engaged. It is tightly engaged with each other, and it is under a high tension. So when it is more and more engaged, the tension will become higher and higher and higher. All right. So if the tension is becoming higher and higher, then if I snap it over there, it should recoil. Okay, so can I measure this? All right. And this can be done by what is called as a laser ablation experiment. And there are nicer ways of showing this, but right now this is shown by, taking any one edge of the cell. That one edge of the cell, we are cutting it essentially with a 2-photon laser in this case. The laser has a wavelength of 800 nanometers, and we can focus it very, very pointedly in this spot. And what you see is that the edges, so basically we can talk about these vertices. These edges are moving away, and what we can count is how much are these vertices of the cells. So essentially this vertex, so if I cut this right here, how much is this vertex moving away from each other, and what is the velocity at which the vertex is moving away? And you can imagine if the velocity is higher and higher, then you will basically come to a point where you can talk about how much tension is there in that membrane. All right. So how much tension is there when it is a circle, and how much tension is there when it is a polygon? Now it turns out that when we do this again and again and again, when it is a more circular membrane, then what we see is that there is some difference, but it's really not significant. But when we have when it's just transitioning to become a polygon, a point at which the myosin is also more engaged, which you saw from the photobleaching experiment, you see that there is more tension and the membrane moves away from each other much more as compared to in this scenario when it is more circular. Right? So this is another way of saying that there is a reorganization of the cytoskeleton such that the myosin is more engaged, and it is engaged to produce a greater amount of tension in this circumstance right here. Okay? And finally, what I'll leave you with is a different set of... So essentially what's happening here is this, right? So what we are seeing is that this shorter membrane correlates with a more circular appearance, and a myosin cytoskeleton which is engaged at some level when it is at a higher, when there are more polygons, there is a greater presence of cadherin, but also that myosin is engaged to actually stabilize this structure. So there's some sort of threshold or some sort of value of myosin adhesion which gives rise to this, some sort of value of myosin adhesion which gives rise to this. And we have a handle on that by just imparting the properties of what is the organization of the cytoskeleton. Now, I'm not showing you these experiments, but what one can do is really decrease myosin or contractility in the system and really increase myosin or increase contractility in the system, right? So over here, for example, there is some change in contractility, but it's a very measured change. It is balanced with adhesion present over there. Now, if I really increase contractility right here, you can imagine that it will never actually form any adhesive structures. So we can do this with mutants. And what we see here is that this structure does not become polygon anymore. Okay, when we really increase myosin contractility, we can do that by activating myosin a whole lot in the system. So it's just essentially contracting in an uncontrolled manner. So that is not allowed for the shape change. The shape change will not occur. And we can do the reverse process where we really reduce myosin activity by using again different kinds of mutants which will remove myosin activation. So now myosin is not getting activated at all. And what happens is an interesting thing right here. So when the membrane is circular, when we reduce myosin further, what we start seeing is the hallmark of polygonal architecture come about at exactly this point. All right. So this change in structure, and it's also known in many different systems that the change in circularity to polygonality or in adhesion to contractility occurs because of very systematic reduction in myosin contractility and systematic increase in adhesion. And for this system, what we can do is correlate this with respect to the amount of organization in the form of the lateral membrane which is present, because the system automatically gives you that facility of looking at a niche where there are different lengths which are available to you. Okay, so I'll basically stop here and take any questions. You have 5 minutes which you may have. Thank you.
I have a question concerning this, the spirals go up and down, and then the, nuclear cycle 13, it's kind of special because it all stops. But what's the signal to stop this oscillation? Or, is it the density of the nuclei?
Yeah. No, this is a nice question for the field. It has been for decades, I think. One of the ways this is explained is that there is a factor which is most likely present in the cytoplasm, which is now getting diluted enough that we don't have the cycling anymore. So the question that he's asking is how in all of this, all of these cycles, for example, suddenly, and this set of movies highlights it well, there is a stop of division and we essentially get formation of these full cells. So what decides this number of cycle 14 for this to take place? And there's a lot of interesting biology to be understood here. One of the famous hypotheses which has been around is that there are a lot of cytoplasmic molecules here which engage and make this cell cycle occur at the pace that it occurs. And once 13 divisions are over, those molecules have become very dilute. So this halts until new synthesis takes place for the next division cycles to actually occur. So this is called also a point at which, I think Shailaja is going to talk about this when she comes and gives her talk. It's called maternal to zygotic transition, mid-blastular transition. It's a point at which the embryo is getting independent of all the material which was present in the early division cycles, and it's starting to make its own material. And this is the point at which this also occurs. But there are many molecules which have been identified. It's not easy to pinpoint exactly which one. I also don't directly study this, so I won't know the current literature, but this is a very fascinating change which occurs.
Okay, thank you.
The cell division process in the first figure, the first movie.
In this one.
Yeah, is it synchronized in time, space as well? Because it seems to nucleate from one point and then spread to the entire.
So if you carefully look at it, and in some sections you might be able to tell this, there is a wave which occurs from the poles to the center.
Okay.
And but it's a very you can understand this more clearly, that there is a wave which goes from the poles to the center in certain mutants much better than in the control. So in general it looks very, very synchronous, and they are occurring at almost the same time. You can see but if you observe them with a greater time resolution, then you can see the wave propagating from the poles to the center for all of the processes. So this is not only the nuclear division, but also the remodeling of the shape and everything. There is a wave. It's very fast, but it's there.
Also, the villi, does it play any role in signaling, because it was brought up?
Yeah, so, I've been wanting to study this, but we haven't done it right now. So, for example, one of the things which you could ask is, what is all of this architecture and how does this correlate with the morphogens? And maybe that is something I can allude to tomorrow. But one of the things is to try and understand in early stages whether there's any correlation between the cytoskeletal or the cytoarchitecture in general to the morphogens. So some of the interesting things which we know is that, for example, the toll receptor, which is involved in the dorsal signaling cascade to form the dorsoventral patterning, the toll receptor is only present in the lateral membrane. It is not really present, or it's very faintly present in the apical membrane. So what does this mean to the toll signaling? I don't know. Toll is also very much like a cadherin molecule. Its structure is very similar to cadherin. And so it is possible that the villi and the lateral membrane have something interesting to do for the signaling, but exactly what it is not something which is very systematically addressed.
And this oscillation between cadherin and myosin, this goes on until...
So it goes on for those cortical division cycles. Yes. Yeah. And also it's not just in this system. It is seen, the opposing relationship between adhesion and contractility is seen in most places where cells are present in consolidated epithelium to when, let's say, migration happens. So those transitions, these changes occur in most of those circumstances in any system.
Really enjoyed your talk. If one were to cause the cadherins to disengage with each other across the furrow, would that stop the furrow formation from?
Yeah, so we have this very interesting mutant of cadherin, which we looked at a long time back. I think I don't have those movies. It's a mutant of a JM domain, and there the furrows don't form.
I see.
So essentially, they don't engage, that doesn't engage very well, and furrows don't form. So the other thing which we also did in this study, and another one which I didn't have time to go into, is that we removed cadherin as much as we can. It's not easy to remove cadherin. It's really detrimental to the system. So we can only reduce it a little bit. And when we reduce it a little bit, then the furrows or the lateral membranes become shorter. So for sure, there is a large... In fact, also the hexagon dominance goes away. So that it's not a stable array either.
Myosin immobilization might also get depleted.
We haven't looked at myosin immobilization, but yes, it's just that the mutant is so difficult to work with.
Yeah.
That's all we haven't done.
That's why I asked about cadherin disengagement rather than knocking it down, because
Yes.
we won't
yes.
even reach the stage.
Yeah. We should go back and look at those JM domain mutants. We haven't done that.
Thanks.
Thanks.
So you wanted to correlate membrane tension with myosin activity, right? Can cadherin activity can also cause membrane tension in some way, because
Yeah, so you bring me to something which I didn't even reach. I think I finished maybe just half of my talk. So, all of this, of course, has to be related to or will change the amount of trafficking which is occurring in the membrane. And what we've seen is that that when we have we are studying this now as well, but what we've seen is that when we have less endocytosis, and I hope that you are talking about Klatsrin and endocytosis, right? So when we have less endocytosis, the villi on the membrane remain longer and the lateral furrow is shorter. Okay, so that is doing two things. So less endocytosis is allowing for the membrane to remain. Now the other question is even more interesting to me right now, which is that if I increase the tension in the membrane, what is going to happen to the endocytosis? And increasing the tension, I can tell you the impact of increasing the tension already. Increasing the tension increases the villi on the membrane, and most likely that is also correlated with less endocytosis. So exactly what one student is studying right now. And hopefully we'll have an answer to what is the relationship of the membrane tension with trafficking in these stages. For sure. Yeah, it's 3:30.
So,
Yeah.
like you mentioned, that in different plastic cell organization, there are different number of different shapes. Like, so is there any ratio which is assigned to them?
So I'll tell you one thing. I didn't go there. What's happening to our maps? So just, this part of it, we were trying to see what is the distribution of the polygons, right, as these cycles proceed. And we have a fair bit of analysis on this, and we have a fair bit of analysis on what happens when you have really unstable arrays of cells at these stages, which means that if you remove, let's say, cadherin, or if you remove other polarity complexes, what happens? So typically, as the divisions are proceeding, and here the polygons are color-coded in these different colors, and gray is hexagon dominant. So as you go in these division cycles, you can see that the grays are increasing. And the gray increase, it also tells you right here that the class of 6 is now increasing, and that is something which is present in all of these, cycle 12, 13 and 14. So remember that it's also increasing in crowding, right, as it is going. But the cycle 11 is very interesting, where the pentagons and hexagons are sort of similar to each other. And then after 11, we have a hexagon dominance which is taking place. So this is telling us that basically in this arrangement, the way the membrane is organized is maybe moving towards a more stable structure as compared to this. But really, when one looks at a stable structure of something like this, then this stable structure which is present in something like this gives you this cell shape index, which is calculated at this value. Okay, and this is what is seen in most mature tissues. This is not a mature tissue. This is a very plastic tissue. It is really undergoing a lot of shape change all the time. So the cell shape index never reaches that. Even if it has more number of hexagons, it really doesn't reach that 3.8 value. Okay, so one could look at some of these measures to understand how they are distributed. One could also work on this in a purely biophysical way, where one could understand how the geometries which are present correlate with the stability. And if it is very stable, will it be this fast? So this is one of the fastest division cycles known in the animal kingdom. And, the cells are dividing in 15 to 20 minutes. And this entire division cycle till 14 is occurring in a matter of 2 hours. So 13 division cycles in 2 hours. It's really fast. So it cannot be very stable. It is not to its advantage to make it very stable.
We look at the human cells. So interphase and prophase, metaphase, all those phases are long.
They're very long, yeah.
So still is there any study related to it? In the interface, they are in the form of circle. So they remain in the form of circle for few hours or one.
No, don't directly translate this to that system. Because, see, when you are looking at epithelial cell, it's epithelial in nature. And then it needs to divide, so that polygon structure there is turning into circular. It's the opposite phenomena. This one is giving you a set of shape changes which are occurring. What you can learn between this and the mammalian system is how does the transition occur from circular to polygon, and whether there are common principles for the two. But if I look at cell division epithelial cells, per se, there is a polygon that becomes a circle, and then it becomes a polygon back again. Okay, so it is the opposite change. We also study that in a different system, but I've not spoken about that. But you can learn from what is coming out over here to what you could tweak there, and whether and ask about whether the same principles are there or not, and to what extent do they matter.